I am Dr. Manisha, going to teach you the histology of nervous tissue. So, what is histology? Nothing but microscopic study of tissue. Okay. So, now by now you would have been very clear with the general anatomy of nervous system. What is peripheral nervous system and what is central nervous system. Now, I am taking a nerve, the specimen of a nerve, and processing it and seeing under the microscope what are the cells and what are the processes I am able to see under the microscope. We will see. Yes. So, this is the nervous tissue, and the nervous tissue, what is present within the nervous tissue? It is composed of neuron with its connective tissue covering. So, all the cells are covered by connective tissue covering, isn't it? Even in the muscle you might have seen the connective tissue, and all other cells are covered by connective tissue. Likewise, the neuron is also covered by connective tissue. Then the neuroglial cells. These are the two important components. One is neuron with its covering and neuroglial cells. What are these neuroglial cells? These are the supporting cells which supports the neurons, which nourishes the neurons. So, in this picture, this is the structure of a neuron, where you can see the cell body and the processes from the cell body. The short processes are known as dendrites. What is the function of this dendrites? It will receive the impulses from outside and deliver it into the nucleus. And there is a long process known as axon. What it does? It takes away the information from the cell body and delivers to the various regions. So, dendrites, short process, and a single long process, the axon. So, these are the parts of a neuron. How these neurons differ from other cells, how it is differing from other cells. So, these neurons have specialized extensions which I said earlier, the axons and short dendrites. Dendrites which bring the information from other regions to the cell body is the dendrites. And what is this axon, the single long axon? This will take away the information away from the cell body to the other regions. So, this is the function of dendrites and the axon. These neurons, how it is differing from other cells, these neurons communicate with each other through electrochemical processes, and they have specialized structures known as synapses and chemicals known as neurotransmitters. These features are not present in other cells. So, coming to the types of neuron. What are the types of neuron? You know, first is the multipolar, bipolar, pseudo-unipolar or unipolar. So, multipolar, from the name multipolar. So, it has got a single axon and many dendrites. So, it is multipolar. Coming to the bipolar neuron, it has got a single axon and a single dendrite, only two poles. So, it is bipolar neuron. And the next is pseudo-unipolar, or you can call it as unipolar, where only one pole is there. From that pole you can see only one process emerging, that is dividing into two processes. So, unipolar, cell body is there and only one process is present. So, it is known as unipolar. These are the types of neuron. These all things you might have read in your schooling itself. Next, coming to the myelin sheath. What is this sheath? So, this is the neuron, and which part is this? This is the axon. Surrounding the axon there is a sheath. Yes, the blue color structure. So, these are the myelin sheath. What is this sheath? So, these are highly specialized cells in the CNS and the PNS. CNS, PNS means central nervous system and peripheral nervous system. Some highly specialized cells will be running around the axons of both central nervous system and the peripheral nervous system. It will be running around the axons and forms numerous layers around the axon, which is rich in lipid and acts as an insulating sheath around the axon. That is the myelin sheath. So, myelin sheath is formed by some specialized cells which is running around the axon laying many layers around the axon and forming the myelin sheath. So, coming to the myelination. So, this myelination in peripheral nervous system is done by which cell? Cell known as Schwann cells. These are the cells which is responsible for the myelination in peripheral nervous system. In central nervous system, which cell does this myelination? The oligodendrocytes. So, these are the two cells which is responsible for this myelination. So, you can see in this picture stage wise it has been, first you can see a single sheath, then the concentric circles. These are all the myelin sheath which is laid by the cell. In the peripheral nervous system it is Schwann cells, and in the central nervous system it is oligodendrocytes. Coming to the myelination. So, this is the histological picture. Okay. Here you can see the round structure. This is the neuron which is myelinated. So, this black structure is the myelin sheath, and inside is the axon. And here another neuron is there with the axon, but it is not covered by myelin. It is unmyelinated axon. This is myelinated axon and this is unmyelinated axon. So, that is it regarding the myelination. And why this myelination is needed? For the conduction of impulses and even for insulation, because myelin sheath is rich in lipid which acts as insulation. Now, the next cell is neuroglial cells. So, these are neuroglial cells. As I said, what are these neuroglia? These are supporting cells. It just supports the nervous tissue and gives nourishment for the nervous tissue. So, in the central nervous system, what are the neuroglial cells? You can see the ependymal cells, oligodendrocytes which we saw already, because this is the cell which is responsible for myelination in central nervous system, astrocytes and microglia. So, these are the neuroglial cells present in central nervous system. What are the neuroglial cells present in peripheral nervous system? It is satellite cells and Schwann cells which we saw already. This Schwann cells is responsible for myelination in peripheral nervous system. So, these are the types of neuroglial cells. Now, we will see about the nerve and its covering. So, what are the layers of connective tissue which is covering the nerve? From outer layer, it is the epineurium, the outermost layer which is covering the entire nerve is epineurium. Within the nerve, you can see bundles of fascicles. The axons are grouped in various bundles. Around each bundle, there is another covering known as perineurium. So, this is the perineurium which is covering each bundle. Still inner to that, each axons has been covered by another connective tissue layer, the endoneurium. Likewise, in the muscle you might have seen epimysium, perimysium and endomysium, the same thing in the nerve. It is epineurium, perineurium and the endoneurium. So, these are the connective tissue which is covering the nerve. Coming to the peripheral nerve. So, these peripheral nerves are formed by bundles of axons which we saw in the previous picture, which may be myelinated or non-myelinated with the connective tissue covering. Nerve fibers are surrounded by supporting cells, the Schwann cells. These Schwann cells are responsible for what? For myelination. Part of the axon between, so you saw the entire axon, the part of the axon between adjacent Schwann cells are not covered with myelin. So, you saw the entire axon which is covered by myelin sheath. So, in between there are some intervals where it is not covered by myelin sheath. That region we call it as nodes of Ranvier. This helps in fast conduction of impulses. So, this nodes of Ranvier is very important because it helps in fast conduction of impulses. So, coming to the connective tissue covering, as I said, each axon is covered by endoneurium. From inner to outer they are moving. It is endoneurium. Each fascicle, fascicle is nothing but the nerve bundle, is covered by perineurium, and each nerve fiber, the entire nerve which is covered by epineurium, which is the outermost covering. So, these are the three connective tissue. So, now you got an idea what is a nerve and its covering and what are the other cells present. Now we will move on to the histological picture. So, once you stain the nerve and see under the microscope, when you take a longitudinal section, obviously it is present in bundles, isn't it? So, you are able to see the round structures. So, these are the axons which is present circularly. Okay. And here you can see this is the transverse section, obviously you will be seeing the circular axons. Here this is the longitudinal section of the nerve fibers. So, here you can see the wavy long axons. So, these are the wavy fibers are the axons. So, here you can see two nerve bundles which is covered by thin connective tissue layer, that is the perineurium, and outside the bundles you can see a thick connective tissue layer, that is the epineurium. So, epineurium connective tissue is very thick, whereas perineurium connective tissue is very thin. This is the picture showing the cell body. This is the cell body of a neuron, and you can see various processes, could be the dendrites, and the one long process is the axon. And along with that you can see small round structures, these are neuroglial cells. Okay. And here you can see the processes from the dendrites and the axons and the center cell body, where you can see the nucleus present inside. So, here you can see the entire optic nerve, and you can see its coverings, and the feature, the identification feature for the optic nerve is, so in this picture it is very clear. You can see again the nerve bundles, likewise in peripheral nervous system you can see nerve bundles, that is the axons which is separated by the connective tissue layers, yes. But here in this optic nerve in center you can see the central artery, you can see the central artery in the center and the central vein of retina. Central artery of retina can be seen in the center and the central vein is seen. So, that is one of the difference between a slide of peripheral nerve and the optic nerve. And one more difference, it is covered by, all the peripheral nerves are covered by only connective tissue, right. But here additional coverings are there around the optic nerve, is nothing but the meninges, dura mater, arachnoid matter and pia mater. These are the three layers of meninges which is covering the optic nerve, because optic nerve is nothing but the prolongation of the eyeball, isn't it? The last layer of the eyeball, it elongates to form the optic nerve. So, it will gradually acquire the coverings of the brain, that is the meninges. Dura mater outermost, the next is arachnoid and the innermost is pia mater. This is the difference between the peripheral nerve and the optic nerve. This optic nerve will be covered by the meninges with the central artery of retina in the center. Remaining all other features are same. So, we will see its myelinated axons grouped as bundles or fascicles, and in center you have got the central artery of retina and vein is seen, and it is covered by three layer of meninges. Here in all other peripheral nerves, the axons are myelinated by what cells? In peripheral nervous system, by Schwann cells. Here it is oligodendrocytes. So, myelination is done by oligodendrocytes in the optic nerve. So, coming to the difference between optic nerve versus peripheral nerve, which is often asked in the viva, in histology viva. So, this optic nerve is covered by three layers of meninges, whereas the peripheral nerve is covered by only connective tissue coverings. And here the myelin sheath surrounds the axons, which is made up of oligodendrocytes. Here it is made up of Schwann cells. So, these are the two important difference between the optic nerve and the peripheral nerve. And one more important feature is, in center of the nerve you can see the central artery of retina and vein in optic nerve. Yes, so till now the peripheral nerve and the optic nerve is over. Now we are moving on to the histology of ganglion. What is a ganglion actually? Before going to the histology part, you should know what is the ganglion. Ganglion is nothing but collection of cell bodies. Okay. These axons and dendrites comes under nerves. These cell bodies collectively known as ganglion. So, what is the definition of ganglion? It is nothing but collection of cell bodies. So, we are going to see about the histology of dorsal root ganglion. So, it is what type of ganglion? It is a sensory ganglion, and where it is present in the dorsal root of spinal nerve. So, spinal nerve has got dorsal root and ventral root. This ganglion is present in the dorsal root of spinal nerve. It is a first order neuron and it has got a thick connective tissue capsule. And what type of neuron is present in this dorsal root ganglion? It is pseudo unipolar neuron. So, these pseudo unipolar neurons, how it looks like a large cells with central nucleus arranged in, and these ganglion cells will be arranged in groups, and satellite cells are prominent. So, surrounding each axons we saw already the satellite cells. Here also satellite cells are present, which is very prominent and helps in nourishment and insulation. So, this is the histological picture of dorsal root ganglion, where you are able to see round ganglion cells. Yes, these are actually, it is not scattered, it is grouped in bundles, and surrounding that blue color dots are seen, those are the satellite cells. And what type of neuron is present in this ganglion? It is unipolar or pseudo unipolar. So, what do you mean by unipolar? I have said already, a single body with the single process, single axon. So, this is the unipolar. So, this is a magnified view where you can see bundles of ganglion, isn't it? Bundles of cell bodies surrounded by satellite cells, and you can see thick connective tissue capsule which is separating it, thick connective tissue capsule. And if you see, the satellite cells are very prominent in the dorsal root ganglion. Yes. So, next ganglion is sympathetic ganglion. So, it is located along the sympathetic trunk and it belongs to autonomic nervous system, and it has got thick connective tissue capsule, whereas dorsal root ganglion has got thick connective tissue capsule. And here what type of neuron? It is multipolar ganglion cells. There it is pseudo unipolar, here it is multipolar ganglion cells. The ganglion cells are small in size, there it is very large in size, and the nucleus, they are centrally placed, here it is eccentrically placed. Eccentric means what? Towards the periphery. And here the ganglion cells are not arranged in groups, it is scattered between the nerve fibers. It is scattered, it is not in groups. And few satellite cells are seen. There more satellite cells and it is prominent also, but here it is only few satellite cells. So, here this is the histological picture of sympathetic ganglion, where you can see the ganglion cells which is scattered and it is smaller in size compared to dorsal root ganglion. It is very smaller in size. This is a hand drawn picture and this is the real picture, and here you can see it is multipolar neuron. Okay. You can see many processes. So, it is multipolar neuron, and the satellite cells are also very less prominent over here. What is the difference between spinal ganglion and the sympathetic ganglion? Very often asked question in the histology discussion. So, spinal ganglion, sympathetic ganglion. So, spinal ganglion is made up of large pseudo unipolar neurons with the central nucleus and surrounded by prominent satellite cells, whereas sympathetic ganglion, small multipolar neurons with eccentrically placed nucleus surrounded by few satellite cells. And this spinal ganglion, the nerve cells are arranged in groups, important point, and in sympathetic it is scattered and it has got thick connective tissue capsule, and here it is thin connective tissue capsule. This is the basic difference between the spinal ganglion and the sympathetic ganglion. Yes. So, by this we are completing the histology of nervous tissue. So, basically when you take a nerve and see under the microscope, what are the features? You can see the neuron and its connective tissue and the supporting neuroglial cells. So, we saw in detail and we saw the slides of peripheral nerve, optic nerve, dorsal root ganglion and the sympathetic ganglion, and we saw the difference between these structures also.